Welcome to Chapter 1, the Introduction to Biology. This chapter is going to outline a lot of concepts that we're going to touch upon multiple times throughout the semester. So don't feel like once we talk about them, that's it. No, we're gonna repeat these things a bunch. It's going to be a kind of a wax on, wax off approach to learning about science and biology. So let's get started, and first let's talk about what life is. How do we define life? What makes a living thing, a living organism? There's a lot of different ways that we've described life before, and we're going to use a few characteristics. You might be able to find other characteristics, and kind of the fundamental reason why it's so difficult to nail down exactly what life is that life is very diverse. It has a lot of different characteristics, and there's some gray areas even when you talk about things like viruses. So let's get into some of these characteristics and get some examples going. The first characteristic of life is that life is organized. Atoms make up molecules, molecules make up cells, those cells make tissues, and it continues. As an example of that, let's look at this plant, Arabidopsis thaliana. We'll just look at a kind of microscopic view of its cells, and you can see that in these cells there's even smaller units called organelles. So the cell is made up of organelles, and then it's embedded within a matrix of other cells, and you can see those other cells are all around it. Well, all these cells together make up a tissue, and those tissues sometimes make up structures. Now you can see in the center we have a bunch of cells making up a tissue, which is making up a structure called a root. And the root system on a plant is actually an organ. So here you can see kind of three levels of structures going on at one time. And finally, all of the organs together make up the organism. Here you can see the shoot system of Arabidopsis thaliana on the far right. And that again is comprised of tissues, which are made of cells, which have organelles, which are made of molecules, which are made of atoms. This pattern of structures making larger structures, which make larger structures, continues on. Let's follow this flow chart, and we'll follow it backwards. And we're starting on the bottom left at number 10. Again, molecules and atoms make up things like DNA. And these structures then, in turn, make up organelles, which are kind of analogous to organs, but for the individual cell. Those organelles are inside the cell and help the cell fulfill its metabolic processes. Those cells together make up tissues. Those tissues make organs, and those organs make up organ systems. So you can think of the cardiovascular organ system is made up of several organs like the heart. All of the organs and organ systems working together make up a complete organism. So in this case, the example organism is this marine iguana. Remember that this marine iguana is composed of organ systems and organs made up of tissues, made up of cells, which are made up of organelles, and ultimately molecules and atoms. Several organisms or multiple organisms in a breeding group, well, we call that a population. If you have several different populations of organisms interacting in a space, we call that a community, and several communities together make up what we call an ecosystem. Now, if we look at all of the ecosystems on Earth together as a whole, well, we call that the biosphere. Let's look at the way that life is organized from a large scale and kind of zoom in and see just how small or how much smaller of a unit we can get to using something like Google Maps. Here I'm on Google Maps, and you can see that we're here in the Bay Area. I've got a bunch of stuff saved all over the place, but I'm zoomed out, and you can see that this is the whole biosphere. There's a bunch of different habitats. You can see there's ice habitats, there's tundra habitats up here. They're very cold a lot of times, a lot of parts of the year. There's these taiga forests up in the north, and there's also these dry areas, these massive deserts. So we can already see that this larger biosphere is made up of these smaller units, these smaller ecosystems that are spread all across the globe. Now let's zoom in and see if we can't see something smaller. Now even on this scale, you can see that the Amazon River goes through this tropical rainforest habitat, this wet forest habitat. And we can see that the Amazon River is flanked by a bunch of green. Now that green is the product of an organism. So we can already see that this ecosystem is made of populations. We'll say these are populations of trees, and those trees are organisms. So we can already see 3 or 4 levels down on the structure. Now, what do you think those leaves, and we're just going to zoom in on the Amazon River here and try to see some up close trees. What do you think that tree's leaf is? Is that an organism? Well, no, it's part of that broader organism. Well, the leaf is part of a shoot system, which is actually an organ. So we're actually seeing in this forest, we're seeing the surfaces of organs that are part of organisms, which are the trees, that are making up populations of trees and a bunch of different species of trees together. It's making this whole community, this whole ecosystem, and this ecosystem is fitting into several other ecosystems around as the conditions change. And finally, all together, they make up a biosphere. So you can see that it's kind of a continuous flow here. The biosphere is made of ecosystems, which is made of communities, which is made of populations, which is made of organisms. And you can really stop at any one of these points and explore scientifically the different structures and components and processes that contribute to these stages. So you might be a scientist that really focuses on DNA, which would be number 10 in this flow chart. Or you might be a scientist that really focuses on tissues and the interactions of different tissues within the body. Or you might be an ecologist that looks at animals on a population and community level. So there's a lot to look at, but all in all, the pattern here is that there's a lot of different organization happening. Another characteristic of life is energy use. A light example of that is a kitten uses energy from its mother's milk to fuel its growth. Plants use light energy to fuel their growth, and this is all part of a metabolism. Another characteristic of life is the maintenance of internal constancy. We call that homeostasis. Within our body, we have kidneys, which help us regulate our body's water balance, and that helps maintain a consistent internal state in our bodies. Here's an example from a paper about heat dissipation in hummingbirds. You can imagine a hummingbird is using a ton of energy flying around and hovering. Well, while it's running its metabolism at such a high rate, it creates a lot of extra heat. In order to maintain a consistent internal temperature, it has to somehow get rid of all that extra heat that it produces. And this paper really illustrates how it does that in a very clever way. You see that we're looking at a hummingbird in flight using an infrared camera that's looking at the heat dissipation on the body of the hummingbird. And you can see a lot of that heat is dissipated through the orbital area around the eye, also in this axial area, kind of like the armpit of the bird, and also on its feet. So next time you see a hummingbird hovering, you can pretty much assume that it's got a lot of heat leaving the eye area, the armpit area, and its little hummingbird feet. Another characteristic of life is its ability to reproduce, grow, and develop. Here you can see a bean going from a seed, sending a root down using energy from those cotyledons, those tissue storages within the seed. It's using that to fuel the growth and fuel these new root tips that grow out laterally. Once those root tips have entered the soil, they can gather nutrients and gather water and further fuel the growth of the seedling. Now this organism is transitioning into a new stage. It's left the soil, and now its first leaves are emerging. And you can see that those leaves are now able to access light and gain some energy from light. It's also at the same time taking in CO2 and using that gas, turning that into carbohydrates and turning that into cellulose, and using that to build new growth, new shoot structures, and it's growing into a new life stage. And so this whole process is an example of growth and development. And you can see that during the time we've looked at the seedling, this sped up footage, it's gone from just a single seed to two developed organ systems. You can see the root system developed, and you can also see a shoot system now developing. Another characteristic of life is its ability to evolve. An example of that is bacteria and its ability to quickly evolve resistant to antibiotic drugs in a lot of different environments. Because bacteria reproduce so quickly, you can actually visualize this with a clever experimental setup and watch evolution and watch the flow of evolution over a well set up petri dish. And what you have is a huge plate of agar, and it's set up in a lot of different bands. And we have basically 10 different bands. On the edges, we have agar that has no antibiotic, but then we have a band inside that has 1 unit of antibiotic. Then we have 10 times more antibiotic, 100 times, and finally at the center of the plate we have 1,000 times higher concentration of antibiotic. So it should be more and more difficult for this bacteria to grow across this plate. Now remember, we're starting from the edges, the right and left side. You can see that these bacteria quickly colonize the antibacterial free area. But then at some point, one of those individuals is a mutant, and it has the ability to resist the antibiotic. You can see there's a lot of different individuals that start to make little populations of mutants, and it continues to grow towards the center. So what you're seeing here is the flow and the selection of individual bacteria that have a resistance to this antibacterial drug. And finally, we see that in the center plate, we have the selection of mutants that are able to resist this 1,000 times potency antibiotic. Now here we've placed dots on where these mutations occur, and we can actually see the flow of mutation and evolution through this bacterial population. So these are the characteristics of life that I'm asking you to remember and study. But of course there's more, and different scientists have different definitions of what life is and what life isn't. What's really interesting is to look into the debate as to whether or not things like viruses are alive. Viruses don't have the ability to generate their own energy. They usually have to steal that ability from their host. And so viruses are an example of something that has genetic material that does evolve, but maybe isn't alive or is alive. So let's move on now and let's discuss cladistics. This is going to be your introduction to cladistics. This is something that we're going to talk about a lot in this class, and I ask you to be able to read evolutionary trees. But as an introduction, let's go through a non-biological example so that we can all make sure that we have a pretty good foundation of understanding how we make these trees, these cladistic trees. So you can see I've set this up to be kind of like a timeline. On the very left you have evolutionary time. On the bottom of the screen is the past, and as we go up we go forward in evolutionary time until we get to the present day. We're going to look at this example organism. Well, in this case it's not an organism, but it's a telephone. So we'll say this is the common ancestor telephone. We know there's a lot of different telephones now, but this is, let's say, where we're starting as far as what the original telephone is in this example. I know there's some older examples, but this is the one we're going to start at. It's like a 1960s rotary phone or something like that. On the far right, you can see there's a blue box, and that's called a state change. And whether or not the state change is on the tree, it's implicit in the branching. That will be more clear as we go through this example. So let's look at this telephone common ancestor. Remember, this is a rotary phone, so to dial it, you have to stick your finger in the number hole and like rotate it, and it's analog. This thing really operates off of just like little mechanical parts. But at some point, somebody introduced an electronic circuit that would automatically make the tones that were required in order to make a call. And so that electronic circuit is kind of similar to what we still have on a lot of what we call landline phones. So you can see that's a state change. That's a change from the rotary phone to the push button landline phone. That happened in the past sometime, and to this day we still have landline phones, even though they're not terribly common. We still have landline phones, and you can push the button, but when you push the button, it goes through a little electronic circuit, and then it makes the call that way. Now, you can see that the landline phone is lined up with the present day mark on the very left in evolutionary time. That's to suggest that landline phones can be found today. So they're what we call extant. That means not extinct. Another state change that occurred sometime in the past was the development of wireless cell phone towers. So these cell phone towers enabled us to have phones that worked not connected, not plugged in. They worked away from these landlines. So we call those cellular phones because they work off that cell phone tower system. And of course, today we still have cellular phones. That probably doesn't look like a cellular phone that you use today, but 15, 20 years ago, that was a cellular phone. So the state change that occurred between that rotary phone and the cellular phone, there's two state changes. One, we went from this rotary phone to an electronic circuit, and then we started using these wireless cell towers, right? Well, there was another state change where we installed these phones into our cars. And you may or may not be familiar with this, but we called this a car phone. And this is actually, I think it's illegal now to be driving around yapping on your phone, especially like a big clunky phone like that. But there was a while where people had these phones installed in their cars, and that's not a thing anymore. So we'll say that that's extinct. So you can see that this line doesn't continue to the present day. It stops. That's because that cell phone technology is extinct. And we'll put that there. The car phone is extinct. Now, sometimes you'll see a little cross next to the name of an organism if it's extinct. In this case, we'll add that. But indeed, the car phone is extinct. Now, the extinct car phone has three state changes from that telephone common ancestor. As an electronic circuit, as a wireless cell phone tower, the ability to communicate through radio waves. And then it also has the installation in a car that makes that its own unique evolutionary branch. Well, let's look at a last branch here, and we call that a smartphone. Now, that's wildly more popular than these other phones at the present day, but you can still find cellular phones and you can still find landlines if you really look. But the smartphone also has three state changes. Remember, it's not installed in a car. So the car phone and the smartphone both have three state changes. The state changes for the smartphone are that it has an electronic circuit, it has a wireless cell phone tower, and it also has the ability to do computing. It's a mobile computer. So in this way, we're going to look at organisms throughout the semester, and we're going to be looking at trees like this, evolutionary trees. And they may not be oriented bottom to top, they may be side to side, they may be top to bottom, but the idea is that the beginning of the branches is a common ancestor. It's an organism that's further back in evolutionary time. And remember, evolutionary time isn't necessarily scalable, it's relative, right? So here we have an example of evolutionary time that's over the time span of maybe a couple hundred years. But sometimes we'll look at some of these trees and the evolutionary timeline will go back millions of years. Let's look at an actual example of organisms in a cladistic tree. Here you can see this is presented left to right rather than bottom to top. And you can see on the far left is the ancestral bear, the common ancestor to all bears. And then we can see a lot of branching. Well, the assumption in this is that at each one of those branches there's a state change, and that won't always be explicitly described. It won't always say it, but that's kind of implicit, and that state change might be genetic. It might be something that you don't visibly see outside on the outside of the animal, or it might be something morphological, like maybe the development of wings in a bat that separates it from a lot of other mammals. The purpose of these trees is to illustrate the relationships between different organisms. You can see here that the polar bear and the brown bear are each other's closest relatives, and those are on a branch that's sister to this branch that includes the Asiatic black bear, American black bear, and the sun bear. And so looking at this tree, you kind of have a way to organize these organisms and understand how they're related to each other, and also to understand how unique some of these organisms are, like the panda bear. The panda bear is on a branch all on its lone self over there, and all the other bears are on another branch. When we describe taxonomic groups, we also use this system of nomenclature. It starts broadly and it goes to more specific groups. So the example that you've probably learned over and over throughout your schooling is domain, kingdom, phylum, class, order, family, genus, species, and subspecies. And those are groups that go from broad to more specific. So if we're using ourselves as an example, we're in the domain Eukarya, we're in the kingdom Animalia, because we're not plants or fungi. We're in the phylum Chordata, that is, we're chordates. And within the chordates, we're in the class Mammalia, so we're mammals. Within Mammalia, we are primates, but there's a lot of primates, remember. But within the primate family, we are hominids. And the group hominids has a lot of different species, most of which are extinct. Our family is Hominidae, which includes the great apes, orangutans, chimpanzees, bonobos, gorillas, and our genus, which is Homo. Our species is Sapiens, and our subspecies is Sapiens. So if you were to describe our genus, species, and subspecies to somebody very specifically, you'd say that we are Homo sapiens sapiens. Lastly, we'll look at another example to really hammer it home. And here what we're doing is we're cutting down species within these groups. We're cutting down organisms till we get to the most specific. So we'll start with domain on top. So the domain Eukaryota, it encompasses a lot of different organisms, from the dogs to the wolves, coyotes, all the way to the paramecium and trees and mushrooms and things like that. All of those are eukaryotes. But if you talk about Animalia, which is one level down, you're cutting out the trees, which are plantae, and the paramecium, which are protists. So you're getting more specific as you look at the kingdom. Well, if you look at the phylum Chordata, you're then cutting the group down even more. You're getting rid of the earthworms and the moths, the annelid worms, which are arthropods, and the moths are arthropods as well. So you're getting more and more specific. And as you go down, you'll see that we're removing more and more organisms from this group until finally, when you talk about a species, Canis lupus, you're talking very specifically about a small group of organisms with a limited diversity as you talk more specifically. And it's no coincidence that the word specific sounds a lot like species. Finally, you have the species Canis lupus, which encompasses the wolf, and the domesticated version of the wolf, which is the dog. The tree of life includes three main branches. On that previous tree, I said that we are eukaryotes. That makes us distinct from these other groups. And we'll look at those other groups now. So we're eukaryotes. There's also another branch called archaea, and then another branch of bacteria. And this really makes up the three main groups of living things on Earth: eukarya, archaea, and bacteria. In later chapters, we'll look at a timeline of life on Earth, and from that perspective, you'll see that archaea and bacteria really are dominant in life on Earth. But it's only later in the evolution of life on Earth that you have these macroorganisms, these bigger organisms, these multicellular organisms like us. But for a long time, our world was a bacteria and archaea dominated landscape. Bacteria are defined as mostly unicellular organisms that lack cell nuclei. Archaea are similar in that they are also unicellular and lack cell nuclei. They're a little different though, and we'll go into the differences later in other chapters, but they do make up about a quarter of the biomass on Earth. So this is by no means a small group. And lastly, the eukaryotes are the group of life that often steals the limelight from everything else because a lot of them are multicellular and they're very interesting. They contain a cell nuclei and they include both unicellular and multicellular organisms. Let's look at the eukaryotes in a little more detail here and kind of look at how we fit into the eukaryotic branch. So remember, we're going from more broad to more specific. We're going from the domains to kingdoms. And one branch of the eukaryotes are the protists, and those are unicellular. Those are kind of the Swiss Army knife organisms that have a bunch of organelles inside them, and they do everything in one cell. And we'll talk about those in later chapters. You can watch them under a microscope, and they're all around. And they really are little Swiss Army knife organisms. The next branch is Plantae, and those are the plants. Those are also eukaryotes. And another branch is the fungi, and you can see here that we're actually more related to fungi than we are to plants. And lastly, we have Animalia. All these groups we're looking at are extant. They all exist today. You can find archaea, bacteria, protists, plants, fungi, and animals. You can go on a walk and find them all. So this layout here isn't totally correct in the way I've laid it out, but what I'm trying to illustrate is all the different diversity that we might be familiar with here on the eukaryotic branch. Let's briefly look at the domain eukarya so you can have an idea of what organisms make that domain. So protists are multiple kingdoms, and they're mostly unicellular. Some are multicellular, some are colonial. Protists can be autotrophic and heterotrophic. So autotrophs would be generating energy from things like sunlight or chemical energy, and heterotrophs would be organisms that have to eat other organisms or matter in order to generate energy. Fungi are mostly multicellular, and those are heterotrophs, so they have to eat things in order to metabolize. So some fungi live in the soil, and they have a co-evolutionary relationship with plants to help them gather more nutrients from the soil. Some fungi are parasitic. Some of them can live on your skin and cause infection. Some fungi grow fruiting bodies like mushrooms, things you can eat. And fungi are a really incredible group of organisms that we don't always notice because a lot of times they're out of sight, but they do make up a large amount of biomass and diversity. Plantae make the plants, and most plants are multicellular. We'll discuss plants in detail in later chapters. Plants are autotrophs, so most plants generate their metabolic energy using sunlight. There's a few tiny exceptions like parasitic plants and we might talk about that more later, but the overwhelming majority of plants are autotrophic and use their chloroplasts to turn light into energy. Lastly, there's Animalia, which is a multicellular group of organisms. They're heterotrophic, so they have to eat and ingest other things in order to metabolize. And it's a very diverse group. You can see here there's a cnidarian, deep sea things. It's hard for me to think of something more different than myself than these deep sea comb jellies and things like that. We'll look at Animalia in further detail later. And this is where we'll be looking at different tissues and organ systems. We'll spend a lot of that time within Animalia. Now let's discuss how science works. Right now with COVID-19, there's a lot of discussion about science, and it's definitely front and center, as well as the relationship between scientists, and the general public, which I would argue are exactly the same thing. But let's talk about all the different disciplines that make up science. And you can see here this little example. There's a lot of different things that are science. Of course, there's math, there's the study of astronomy, there's the study of geology, there's the study of chemistry and technology. These are all different sciences. And some people divide those up into life sciences and applied sciences. Indeed, there's a lot of ways to look at all of the different scientific disciplines and to divvy them up. This chart for me is kind of confusing, but I want you to notice on the left side that we're trying to organize science on a scale level. So looking at things on a small scale versus on a larger scale. And you'll see that kind of within this, between atom and Earth-size scale, we have the life sciences, and there you'll find functional biology and cellular biology. You also find the social sciences and psychology, and these are all things that are rooted in living things. Remember, we defined what a living thing is. Well, the study of living things and kind of the consequences of living things all is embedded in that scale between the atom and the solar system. You find that scale. And so we're really going to be looking at the life sciences this semester. The physical sciences and the formal sciences, you can see, are very important, especially at the tiny scale, below the scale of the atom. And also on these larger scales, as we start looking at the solar system and the Milky Way and just the entire universe, you have to really root yourself in math, physics, and your understanding of physics. And so these are all sciences, but what's something that's helpful for me to keep in mind is that you don't have to be an expert at chemistry and physics to learn a whole lot about biology. And you don't have to be a mathematician to gather some insight into the world using physics. A lot of this stuff is a little more intuitive than you'd think. And that's what we're going to try to do this semester. We're going to try to give you the tools. I'm going to try to give you the tools to learn about biology and learn about science without necessarily being a chemistry whiz or a physics whiz or a math whiz, because I certainly am not any one of those things. So this is a great example of all the different sciences that exist. They certainly do play an important part in our daily lives. Technology is an example of applied science, but there's a lot of different methods that we use, different methodologies we use to understand the world in a scientific way, and that's what we're going to talk about next. But before we go into that conversation, I'd like to first kind of prepare you for an understanding of how we look at the world, and to kind of take note of a potential bias in everything we discuss. I think this is an important conversation because as we look at our understanding of the world, it's important to look at the filters and the channels that information had to go through. And then we can think about how we might change that in the future. And so What do I mean by that? Well, on the left is the distribution of the top universities on Earth. So just think about this from a biological perspective. If we're trying to understand life on Earth, we're trying to understand living things, this is where all of the science goes through, right? It goes through the universities. And you can see that they're heavily biased towards temperate regions. They're heavily biased towards Europe. They're heavily biased towards the East Coast and the coasts of the United States. And there's some hot spots there, and also on the coastal areas of China and in Australia. These are all temperate regions. On the right, you can see an example of all the diversity of planet Earth. And you can see that the most diverse places, which include the Amazon basin, which includes equatorial regions of Africa, these are the places with the most speciose habitats, the most species of amphibians, the most species of mammals, the most species of birds. These are the places that have the most life. You can see here that these areas that are hotspots for animal diversity, indeed, if you wanted to understand life on Earth, this is probably where you would want to be. There are also these places that are devoid of institutions like universities and higher education. Now that we've illuminated this potential bias, let's continue talking about science. This was just something I wanted to mention early on to kind of give you some tools when framing how we view a lot of different things. And I might touch upon this pattern in future lectures and different times, and I'll usually refer to it as a temperate zone bias, which is likely why we know so little about so much of biological diversity. So let's talk about the kinds of reasoning and the kinds of thinking that go into the framework of science. So the first thing we want to talk about is inductive reasoning. Inductive reasoning is a process of observation. It's a way of looking at the way things are and thinking about how there's a pattern and observing those patterns and documenting those patterns. It doesn't necessarily mean you're doing an experiment, but this is an important step and stage and process in science. In this example, you have Jane Goodall, a famous primatologist. She's observing chimpanzees, and she's not doing an experiment necessarily. She's not performing replicates of this moment or anything like that. She's simply taking notes and generating ideas and observing what's happening before her. And this is actually a very important stage in science, and even in experimental science, you're often going back into this stage and going back and forth. So let's look at another example of inductive reasoning. And this is actually a published paper. This is a descriptive paper. It's a journal article talking about the luminescence of a millipede. So someone observed that these millipedes glow in the dark. They're glowing at night, and they took notes and they observed this. And they even, on the top right, you can see they even measured the wavelengths that were reflecting off of this millipede. They didn't necessarily do an experiment, but really this is an example of inductive reasoning, the observation of what's happening in nature, and just the documentation of that. Now is a great opportunity for you to think about this scenario and to make some guesses as to why this millipede glows in the dark. And that's when you'll transition from inductive reasoning to deductive reasoning. So let's say you think that this millipede glows in the dark because perhaps this is a way to warn organisms not to eat it. Let's say this is a warning color. You think that this is a warning color. In order to show that this is a warning color, you would have to make an experiment. And so now is an example of a paper that is using deductive reasoning to try to explain why these millipedes glow in the dark. And this is a great example because it's a very simple experiment. And the hypothesis was that the luminance, the glowing, was to warn predators that this millipede is toxic. And this millipede is toxic. It produces cyanide, so it's not a good thing to eat. So what they did was they made clay models. These researchers made little clay models of the millipede, and some of them they made them glow in the dark, and some of them they didn't. And they put these clay models out, and they let them sit there overnight, and then in the morning they went back and checked to see if there's any bite marks on the clay models. The bite marks would suggest that a rodent or some predator tried to bite it and tried to attack the clay model. And what they found was that the predators were biting on the non-glowing millipedes. Well, using deductive reasoning, they suggest that predators won't bite the glowing millipedes because they think that the glowing millipedes are toxic. This is an example of deductive reasoning stemming from inductive reasoning. And whenever I show you examples like this, I'll often include the paper on the bottom right or the top left or somewhere in the corner, and you can look up that paper and read it on your own time if you're interested in this stuff. And this is a really cool experiment, super simple, something that, this is something you could conceivably be involved with, as a graduate student in just a few years. So this whole process makes up a framework that we call the scientific method. And the scientific method is something that we present as a kind of a flow chart, a very organized process, but it really is kind of messy. And when you start doing experiments yourself or looking into how these things really evolve, a lot of times it's just a sloppy back and forth. But generally speaking, it goes like this. You make an observation. Oh, these millipedes, they glow in the dark. Then you ask a question. Why do they glow in the dark? Then you form a hypothesis that answers that question. I think they glow in the dark because it warns the predators that they have cyanide in their bodies. Then you make a prediction based on the hypothesis, which is I predict that predators will attack a non-glowing clay model more than a glowing clay model. Then you do an experiment to test the prediction. You make the clay models, you go out, you do different replicates, and you have all of your science friends help you out. Then you gather the data and you analyze the results. Those results either support your hypothesis or do not support your hypothesis. Then you report the results in a scientific paper. That paper goes to a journal, and we'll talk about that step next. But if the hypothesis is not supported, you can see that it still goes and you still report it, but then you try to find the reason why. What's important in this process is that you're letting nature tell you how it works. You're not telling nature how it works. So you don't make an experiment to prove your hypothesis. You make an experiment that would disprove your hypothesis or allow you to say that it's hard to disprove. It's kind of a weird way of thinking, but basically science is founded on the principle that we don't really have a way to say what's absolutely true, but we can say what probably isn't the case. And so a lot of times when you hear scientists talking about science, they won't ever speak in the affirmative. They'll never say, oh, this is what's going on. They'll say things like, oh, this suggests that this is happening, or it's likely that this is happening. And that's just the way scientists talk, because we can never really prove things 100%. We're always using deductive reasoning to show what isn't the case, but we're always open to the possibility that future experiments could actually disprove what we think is the best explanation. When you do have these results and you want to report them, you report them in a peer-reviewed journal. And there's a lot of different peer-reviewed journals, and these are different than what you typically find in the news. So a newspaper is not peer-reviewed. A lot of what you read, let's say you go to Google News and you read something on BuzzFeed, that is not peer-reviewed primary literature. That sometimes is based off of peer-reviewed primary literature, but it really isn't as thorough and rigorous and as reviewed as these scientific papers that come from these journals. let's look at what makes these peer-reviewed journals distinct from other kinds of media and other kinds of news. When you make an experiment, and this is something that we may do in labs, in future labs, you want to make a simple and direct experiment. You want to test just the hypothesis that you have, and you don't want to inadvertently test other hypotheses. So you want to make sure you're testing one thing at a time, and that can be a little more deceptive than I just said. You also want to have a control, and controls kind of remove variables, and we'll go into that more as we have better examples that illustrate what controls are. You also want to have a good sample size, because if you have a small sample size, your results could be just due to chance. So an example of that would be, let's say you want to show that a penny is 50-50 heads and tails, but you only flip the penny twice, and both times it's heads. Well, you can't say that this penny only is heads up because you only did it twice. You would have to flip that penny like 100 times before you started showing that it's really 50-50. If you did it twice, there's a good chance that you would have an error due to sample size, and you would have heads twice or tails twice. And then you collect the data. So you have to decide what kind of data you'll collect. And finally, you have to analyze that data. When you're finished with your experiment, you write a manuscript and you attempt to publish your experiment in one of these peer-reviewed journals. So the structure is very, very consistent with these peer-reviewed journals, and this is a structure that's been used for a very long time. And it's kind of helpful that the structure is so basic and predictable, because I can read a paper from 50 years ago and I still kind of know how to navigate the paper. It keeps things consistent over time, but it kind of makes things feel a little dry by today's standards, but that will be a consistent way of communicating to scientists in the future. You also make a paper to fit your particular journal. You want to think about what journal you're going to write for. So let's say you're doing this experiment with millipedes, you're probably going to publish that in an insect or arthropod journal. Now, once you submit the paper, other scientists get to read it, and they send that paper out before they publish it for other scientists to review it anonymously and to either okay it and say that it's good science and that it's all checks out, or they say that it's not good science and you did something wrong. And then they'll either reject the paper or they will ask you to revise certain parts of it. And this is a really important step, and this is what makes peer-reviewed literature different from all the other literature that you find about science. Peer-reviewed papers have gone through this rigorous check, and they're not necessarily checking to see if they agree with your science. They're checking to see if you're approaching science from a non-biased perspective, and if your experiment really does show what you're saying it shows, and if you're not saying that it shows too much. A lot of times we extrapolate or we make conjecture about our experiments and say that they're showing more than they really are showing. Essentially, after peer review, your paper is either rejected or accepted. Oftentimes it's rejected, and if it is accepted, it gets published, and then it's presented in these peer-reviewed journals as information that other scientists can access and use, and they can then kind of piggyback off your research and advance what you did and gather more insight and knowledge into the way that nature works. This is the process through which scientists have, over time, built a grand idea of how we understand nature. And this stems from astronomy, our understanding of physics. There's math journals, there's journals on technology, there's journals on biology, and all of these journals are kind of the source material that's used by scientists to then generate new hypotheses and further our understanding. So where do you find primary literature and this peer-reviewed science? Oftentimes through your university or through your college library, you can find a lot of different peer-reviewed papers. Because peer-reviewed publishers are very, let's say, lock and key with access to their journals, it can be a little clunky using online libraries, but Indeed, on DVC's website or on a lot of different college websites, if you're a student, you'll have access to a lot of peer-reviewed literature. You could also go to the library and pick up the actual physical hard copies of these journals, and that's how we used to have to do it. But a lot of this material is now published online, and a really easy way to find peer-reviewed literature is using Google Scholar, which is one of the lesser known Googles, but it is a great resource and really can help you find almost all published research pretty quickly, pretty easily. And a lot of times you'll find a paywall, but we'll talk about how to address that in later lectures. In later assignments, we'll look at this process and we'll do some comparative homework here, and we'll really get a better understanding of how to read this kind of literature. But at this point, I just want you to know that there is a distinct difference in the way that we publish these peer-reviewed scientific journals versus a lot of the material that we find online or in the media that gives us information. So this is a different way of getting information, and it's very rigorous, and it's a very thorough process that is our best way of ensuring that we are building verifiable results upon verifiable results. That's all I have for you for this lecture, and I hope you enjoyed our discussion and our introduction to biology and science.